By the end of this lecture, I hope you will be convinced that our biology is much more flexible than we used to think, and we can bend it in a shape of health or disease, depending on the lifestyle choices we make. This process is called biological learning, and it is based on the ability of our biology to store a memory of our lifestyle experiences in order to adapt to an environment in constant change. We now have the tools to measure biological learning and use it clinically to improve our health and longevity. Specifically, we will learn how genes learn through a process called epigenetics, why food is an opportunity for biological learning, and what are the good and bad epigenetic memories, how we can measure and control them to improve our health and longevity. Genes learn through a process called epigenetics. The Greek prefix epi means on the top. So if our genome is our DNA, our epigenome is a set, a layer of molecular tags placed on our genes that can turn genes on and off. And epigenetics explains why we have different cell types in our body. Liver cells, brain cells, fat cells, all told more than 300 different cell types that all share the same DNA, the same genome, the same hardware. How is this possible? Because each cell has a different epigenome, a different software that modulates the expression of the hardware, the genome and genotype, into phenotype, the way the particular cell identity and function of each type of cell. And that's why epigenetics can explain why different cells and different organisms with the same genome can actually have dramatically different phenotypes. For example, cells, eye cells expressing different color pigments, or identical twins, the caterpillar and the butterfly, and worker bees and queen bees in a beehive. In order to understand the epigenetic software, we need to describe two important properties of the epigenome: epigenetic flexibility and memory. Unlike genetic sequence, our epigenome is flexible and dynamically remodeled by environmental factors we are exposed to every day that make up the so-called exposome. Factors like diet, pollutants, stress, drugs, chemicals, additives, and all these factors send signals to our epigenome and modulate the activity of writer, eraser, and reader enzymes that write, erase, and read epigenetic modifications, thus modulating gene expression. And therefore, you can think of our epigenome as a set of memo nodes that store a biological learning memory of our lifestyle exposures. And this delicate balance between flexibility, responsiveness to the environment, and stability through cell division is the molecular foundation of biological learning through epigenetics, which is called epigenetic memory, and is the ability of our epigenome to respond to the environment and remember our lifestyle exposures. And the memonotes that are placed before our birth tend to be written with a pen and be permanent, whereas the memo notes placed after our birth tend to be written with a pencil and potentially reversible. And both pen and pencil marks contribute to our health and disease risk. Now, food and diet represent the most powerful signals to our epigenome, and that's why food is not only calories, but it is information, as Dr. Jeff Bland once beautifully said. And specifically, if my research could be a painting, it will be this painting here by Giuseppe Arcimboldo. And Giuseppe and I share a couple of common characteristics. We are both Italian, and we both believe that food can shape our biology and our biological learning. Specifically, I'm looking at how a specific type of epigenetic modification called DNA methylation can shape our biological learning in the context of the largest randomized clinical trial ever undertaken to compare a low-carb and a low-fat diet, the Stanford Diet Fit Study by Professor Christopher Gardner, in which 609 overweight men and women were randomized to either a low-carb or a low-fat diet, which they followed for 1 year. And the reason why we are looking at the DNA methylation is because we can quantify DNA methylation with high accuracy and at the single nucleotide resolution level, whereas this is not possible with other epigenetic modifications. And several DNA methylation studies have shown that nutrition can program the ability of the epigenome to modulate the expression of genotype into phenotype through a process known as nutritional programming, which plays an important role, especially in early life development. For example, a very famous nutritional programming study was done in honeybees and showed that worker bees and queen bees in a beehive have actually the same hardware, the same genome, but a different software, a different epigenome, and this difference is programmed by nutrition in early life. So only queen larvae are fed with royal jelly, a protein rich substance that activates an epigenetic program that switches on the genes required to develop the queen bee phenotype. Queen bees are not only larger than worker bees, but they live 20 times longer, and they are fertile, whereas worker bees are sterile. And this is all due to nutritional programming early in life. And we also have our royal jelly. This is what epi-nutrients are. Nutrients that can nourish our epigenome and unlock our full potential. And epi-nutrients can be broadly divided into two categories, methyl donors and epi-bioactive nutrients. Methyl donors provide the building blocks for methylation reactions, which play an important role in several epigenetic modifications, including DNA methylation, but also histone modification and modifications and epitranscriptomics. And methyl donors include a variety of nutrients in plant and animal foods, such as folate, vitamins in the B group, choline, betaine, whereas epi-bioactive nutrients modulate the activity of writer and eraser enzymes, and include an extensive list of nutrients, of which you can see here only a very limited snapshot. For example, T polyphenols, resveratrol, sulforaphane in broccoli, and butyrate, a compound produced in the intestine when dietary fiber is fermented. Another very famous study about nutritional programming is a study done in the so-called Agouti sisters. These two mice are called Agouti sisters, and they have the same hardware. They both they have the same genes, and specifically they both carry a gene called the Agouti viable yellow allele, or AVY, which programs them to develop a yellow fur and a voracious appetite, which eventually makes them become obese and develop diabetes. However, the AVY allele is turned on only in the yellow fat mouse, whereas is switched off in the brown lean mouse. And the reason for that is nutritional programming early in life. Specifically, when the mother of these mice is fed with a standard chow diet, which is poor in epi-nutrients and methyl donors, then a greater proportion of the offspring cannot turn off the AVY allele, which is expressed, thus making the mice become yellow and fat. Whereas when epi-nutrients are added to the diet of the mother, a greater proportion of the offspring can turn off the AVY allele, thus becoming lean and brown despite having a genetic predisposition to become yellow and obese. So nutritional programming really describes that we are what we eat, and perhaps even what our parents and grandparents ate. And there is evidence that nutritional programming may occur even in humans. During World War II, a food embargo was imposed in Holland, and as a result, more than 20,000 people died during the so-called Dutch hunger winter. The famine babies, the babies born during the famine, were born underweight and with several health issues, which is not surprising. What is surprising is that the famine babies had a higher risk of becoming obese and diabetic when they became adult. And even more surprisingly, the children of the famine babies were also born underweight and with a high predisposition to become obese, even if they were never exposed to the famine. So it seems that the famine, the epigenome of the famine babies stored a permanent memory, a permanent biological learning memory of the famine that programmed their genes to change the metabolism to store more calories from the food they ate. And even 6 decades after the famine, scientists found that compared to their same-sex siblings who were never exposed to the famine babies had some distinct DNA methylation changes in metabolic genes that regulate processes like insulin secretion and energy metabolism. And these DNA methylation changes are now studied as epigenetic biomarkers for the prediction of type 2 diabetes risk. Now, the good news is that several lifestyle exposure can induce good epigenetic memories and biological learning. This includes epi-nutrients, calorie restriction and fasting, exercise, stress reduction and meditation, social connection, and a whole food nutrient-dense diet. For example, in our Stanford Diet Fits, we see that both a low-fat and a low-carb diet can induce positive DNA methylation changes. For example, a low-fat diet affects DNA methylation of genes involved in the protection against colorectal cancer. The genes are here marked with a red asterisk. And a low-carb diet affects the DNA methylation of immune response genes, especially as related to natural killer cells, which play an important role in the fight against viruses and cancer cells. On the other hand, we also know that several environmental exposures can trigger bad epigenetic memories, and these include smoking, obesity, environmental toxicants such as plastic compounds, BPA, phthalates, and also pesticides such as glyphosate. Early life trauma, microbiome imbalances, and sleep deprivation. However, the good news is that some of these bad epigenetic memories may be reversed through lifestyle intervention. For example, we have published a review of weight loss studies showing that weight loss can reverse obesity associated DNA methylation and potentially the health damage associated with these epigenetic memories. Now, the burning question is, can we measure biological learning? And epigenetic biomarkers may help us do that. We have seen that epigenetic biomarkers are molecular signature, molecular fingerprints of certain life exposures. For example, the famine-associated epigenetic biomarkers of type 2 diabetes. We are also looking in our Stanford study at some other obesity-associated epigenetic biomarkers for the prediction of type 2 diabetes. And a specific new emerging category of epigenetic biomarkers are epigenetic clocks. They are a sort of a snapshot of our good and bad epigenetic memories throughout our life and can therefore capture the passing of time. Specifically, they can capture both chronological age, which we usually measure by counting the candles on our birthday cake, and biological age, which is the age of our cells and tissues and can differ from chronological age. We can be biologically older or younger than our chronological age. Different epigenetic clocks are more sensitive to different epigenetic memories and aging signals, and therefore there is no best epigenetic clock, but one big idea. Epigenetics revolutionizes the way we describe, measure, and target aging, not only in terms of time, but in terms of biology. And while we can't control time, we can affect biological learning through epigenetic and lifestyle. And several intervention studies show that we can rejuvenate epigenetic age with different lifestyle interventions, ranging from supplementation with vitamin D3 to a Mediterranean diet to a multimodal lifestyle intervention involving a diet rich in epi-nutrients, low in carbohydrates, with some intermittent fasting and stress reduction management. So I hope that you can now see that epigenetics marks a paradigm shift in health. And so from fear of disease to opportunity for health. That our genes are only the starting line in our health journey, and our epigenome is our path to health. And diet is the best epigenetic pencil we have in our hands. And so the take-home messages of this lecture are that our genes can store an epigenetic memory of our lifestyle exposures, thus learning to adapt to an environment in constant change. That food is one of the most powerful triggers of biological learning through epigenetic memory, and that biological age reflects our epigenetic memories can be estimated by epigenetic clocks and can be influenced by our lifestyle choices. If you want to learn more about the science of nutritional epigenetics, subscribe to my YouTube channel and check out my newsletter using the link in the video description. You will get access to free lectures from my Stanford courses on epigenetics, longevity, and nutrition, and be the first to know about my upcoming webinars and courses. I hope to see you there.